Good morning, everyone. I wish I could be there with you today in person for this All of Us Science Day to celebrate the discoveries emerging from this remarkable program and the community of researchers and participants who make it possible. People often ask a simple question. Well, we sequenced the human genome more than 2 decades ago, so why hasn't that translated into cure after cure after cure? Well, the answer is it's translated into some cures, but not every disease is purely genetic. Human health is shaped by a complex interplay of our genes, lifestyles, and environments. Understanding that full picture is essential if we're to unlock the biggest mysteries in medicine. The All of Us research program was founded on exactly this premise, that we need a research resource capable of studying how genetic, lifestyle, and environmental data converge and interact. That three-pronged approach is what makes this program unlike anything else in the world. There's also a paradox at the heart of precision medicine. To tailor treatments to individuals, you actually need very large populations to uncover the patterns that connect genetics, lifestyle, and the environment to health outcomes. And that is exactly what the All of Us provides, research at unprecedented scale. All of Us also demonstrates how research moves from discovery to impact. The program has already returned individual DNA results to more than 277,000 participants, including nearly 5,500 people who received potentially life-saving information about preventable hereditary disease risks like BRCA2. What makes All of Us truly transformative is not just its scale, but it's also the depth of data contained within a single research resource aligned with my priorities for the NIH and designed to address health questions that no single dataset could answer on its own. We're also working toward a future in which NIH research resources connect and interoperate, sharing standards and common data models and analytic approaches to amplify reproducible science and public health impact. In many ways, All of Us is pioneering this approach. Today, the All of Us Researcher Workbench supports more than 21,800 researchers from every state, allowing reproducible, cloud-based, collaborative science at scale. And later this year, they'll be opening up biospecimen access. This will allow investigators to pursue even more ambitious scientific questions using this unparalleled resource. I see All of Us as a national treasure, an accessible, foundational platform that investigators at every career stage in institutions across the country can use to tackle our most pressing health challenges. And within the NIH itself, All of Us has become a powerful shared resource. Nearly every institute and center is exploring ways to build research efforts on top of this platform, saving resources and launching national-scale science in months instead of years. So again, thank you to everyone here today. Many of you are already using this data, and some of you will be presenting your findings. This is exactly the kind of large-scale, rigorous, real-world science that will help solve some of the biggest mysteries in human health while accelerating discovery across the NIH. Most importantly, none of this would be possible without the 876,000 participants who have placed their trust in this program and the researchers who are transforming that trust into discoveries that improve health for every American. Thank you for being a part of this extraordinary effort. The science you'll hear about today is just the beginning. I look forward to seeing what's coming next.
Hello, my name is Min Kyung Lee, and I serve as the Acting Chief Engagement Officer for the All of Us Research Program. And we just heard from Dr. Jay Bhattacharya, Director of the National Institutes of Health. On behalf of all of us and the NIH, I welcome you to All of Us Science Day 2026. Science Day began as a 2-day student researcher symposium in 2022 with 650 attendees to showcase the work of researchers using All of Us data. Now in our 4th year, we're excited to be hosting over 5,000 registered attendees featuring 6 scientific sessions and poster presentations drawn from a competitive call for abstracts. For the very first time, Science Day is open not only to researchers, but also to participants and the public, both across the United States and territories and worldwide through virtual participation. This year's theme, From Discovery to Impact, reflects our shared momentum, uniting researchers, participants, and the public to celebrate discoveries that are driving meaningful change in some of today's most pressing health issues, all made possible by all of us participants and researchers. Before we begin, I'd like to share some housekeeping notes. Science Day is a public event. To protect your privacy, you can change your display name before entering a session. Closed captions are available throughout the day To turn these on, click the CC icon you see in the top right of your screen and choose your preferred language. If you run into any technical issues or need an accommodation, please email allofuscienceday@pyxispartners.co. To ask a question, select the Q&A option at the top right of your screen, type your question and submit it. The chat box will not be monitored for questions, so please make sure to use the Q&A box. However, feel free to use the chat box to share comments, feedback, and reactions. There's also reaction buttons at the bottom. We have an exciting lineup of sessions for you today. As we host a mixed audience of researchers, participants, and the public, please make note of the purple tag as you see here on the agenda, labeled General Audience. These sessions are specially designed to use plain language, while other sessions may be more research-focused and may include more technical detail. Of course, everyone is still welcome to attend all and any sessions as you'd like. And then at the end of today's agenda at 4:30 p.m. Eastern Time, we have our participant forum and closing session. Based on feedback from our participants, we've invited researchers using All of Us data to present on genetic risk, cancer prediction, and how people respond to food, followed by a live Q&A with program leaders. We also have awards to celebrate our researchers. Please make sure to vote for your favorites by 3 p.m. Eastern Time in the abstracts and posters pages of the Science Day website. Awards will be presented at the closing session, so I hope you can stay with us till then. Now, it's my pleasure to introduce you to the speakers in our opening session. First, we'll hear from Dr. Joshua Denny. Dr. Denny is a medical doctor, proud father of 4, and chief executive officer of the All of Us research program. Before leading All of Us, he was a practicing physician and a leader in personalized medicine at Vanderbilt University Medical Center, where he focused on using electronic health records and genetic information to better understand and treat disease. After Dr. Denny, we're grateful to have Dr. Naomi Allen with us as our invited speaker. Dr. Allen is chief scientist for the UK Biobank, where she is responsible for following up with participants, both through linkage with routine health-related datasets and through web-based questionnaires. She's also involved in developing the scientific strategy for future enhancements for the study and serves as an advisory panel member for All of Us. Last but not least, we'll hear from Dr. Jeffrey Ginsberg, Chief Medical and Scientific Officer and Acting Chief Data Officer of the All of Us research program. Before joining All of Us, Dr. Ginsberg founded the Center for Applied Genomics and Precision Medicine at Duke University. Today, he sets the scientific vision and strategy for All of Us and oversees data collection and curation and new data types to drive discovery and innovation, which we'll explore today. Now I'll pass the mic to Dr. Denny.
Good morning, everyone. It is a pleasure to be with you today. I am thrilled that you have decided to spend some time with us today for All of Us Science Day. I'm also thrilled that we have all parts of our community here, participants, researchers, staff, and other program leaders across the country, and public members to hear what impact All of Us is having across the program. I'm going to start, before getting into the science, talking a little bit about my personal medical history and my why as why I'm part of this program. This is a picture from left to right of my great-grandmother, my grandmother in the back, my uncle, and my dad in the front, obviously as kids. But what you don't know here is that there are 4 people that should be in this picture that aren't. My dad and uncle lost 4 siblings that due to a rare genetic condition called cystic fibrosis that had no treatment at the time. My dad recalled stories of taking a sibling to the hospital for the last time. My grandmother devoted her life to betterment of kids, following that, serving as a preschool director for 30 years, and obviously countless hours invested in me and her other grandkids. Cystic fibrosis is a rare recessive genetic disease, which means you need to inherit 2 copies of those spelling changes or mutations in your DNA that cause the protein that causes CFTR, to be misfunctional. And there are about 2,000 mutations, we think, that are causative of CF, but you have to have 2 of those copies. It causes problems in many different organ systems, from the lungs as one of the places people think of most: asthma, cough, thick secretions that result in recurrent pulmonary infections. You have skin changes, you have liver changes, pancreatic changes, intestinal changes such that you can't digest and absorb your food well. You can be infertile. All these things result when, in my grandmother's case, in grandfather's case, very early death. When I was a resident, supportive changes made these lifespan of individuals with CF into the 20s and 30s common. But in 2013, the first targeted medication, the first precision therapy came out, which only worked for 5% of people with CF, specific mutations. But for those people, for the first time, lung function improved, cough stopped, changes happened. You saw a path towards a normal life with CF. And by 2019, triple drug therapy came out that now addresses more than 90% of people with cystic fibrosis. This is the kind of story of precision change and precision therapies that I hope we see across all diseases that I want to enable with all of us. But it also reminds us, and for me personally, that behind every story and every disease we tackle is a family, is a personal story, is our memories of loved ones that we can tackle and that we can make better, not even to mention ourselves. This brings us to all of us and where we are now. Nearly 900,000 of you have joined from across the country. We represent the entire country, really more than any other study in the US. We have 98% of the ZIP3 zip codes represented. That's the level we de-identify those addresses you enter into as the first 3 digits of your zip code. And you can see we represent the entire country. We have most US territories represented as well, rural, urban, many populations. People participate in different ways. 95% of people say that they'll share their electronic health record with us. We have a harder time getting those electronic health records, but we are making dramatic progress with nearly 500,000 electronic health records collected from across the country. We over 600,000 have donated biosamples. That's a minimum of DNA. And we're turning those samples into real translation into real research. The way we do that is through a cloud-based researcher platform where researchers come to us to use the data. It is our researcher workbench, and I'll show a screenshot of that in a little bit. These researchers come from across the world. We have nonprofit, academic, educational, commercial, healthcare institutions using this. We have high school students who have created award-winning science projects with us that look at genetic mutations and risk of cancer, for instance. These hail from about 1,300 organizations, and they've created over 24,000 workspaces and projects investigating all kinds of diseases. Everything really you can think of, from common disease and rare disease, is represented. One of the things by bringing researchers to our platform is we can allow researchers to gain access to our data, the data that our participants have shared, including myself and my family, in a rapid fashion that is safer as well as faster and more democratic, to let more research ideas be explored faster and safer at the same time. Researchers come in after verifying their identity, going through specific training, verifying through an institution as well of who they are, and that the institution vouches essentially for their identity as well. And then they can start doing work. It only takes an average of about 28 hours for a researcher to come into the program if they're part of one of those 1,300 partner organizations to get approved to start doing work. And then we can monitor the work they do through our resource access board and the project descriptions they fill out. One of the things we want to do is we want to take those studies that people do, those papers they publish, and make them visible to our participant and other researcher communities as well. One of the things we've introduced here recently is essentially real-time AI summaries from the papers that are published. So as we put them into our publication directory, and you can see that publications are growing exponentially. We had almost half of our publications that came in 2025, and you can see that 2026 is taking off from there. We produce those summaries so you can see and have them readily available in lay language as well. And so this study here is one that I highlight. What we have noticed is people with Alzheimer's disease with a spelling changes or genetic variants in ApoE gene are 10 to 15-fold increased risk of having Alzheimer's dementia if they have 2 copies of that gene. But we've noticed that people of African genetic ancestry tend to have a reduced risk of having Alzheimer's disease despite this variant. And this study shows that there's a 19 base pair segment of DNA right in front of that gene that is common in people of African ancestry that reduces the risk of having Alzheimer's disease. This is important because it helps us get more precise in understanding risk of dementia. It also could point to treatment modalities for all populations. And we see this over and over in the research that we have published on our resource. Now we have produced quite a bit of data. In dark blue, you see the data set that is available to researchers now, 630,000 individuals with survey data, over 400,000 with whole genome sequences. In light blue, you see the next data set that will come out probably around June or July. That data set will include about 10 to 30% more individuals in every category. In some categories, there's many fold changes in data that we have new data coming out that's never been released before. We'll have over 535,000 whole genomes, nearly 500,000 with electronic health records. The wearable data, which is already the largest longitudinal research wearable collection in the world, will go from 9,000 to about 80,000. The long read data will go to 15,000, including methylation data that will help us think about environmental exposure. And we'll have our first releases of data like proteomic data and transcriptomic data. It looks at RNA data, and then exposomic data. And finally, we will do a first release of extracts from clinical narrative data. A lot of times clinical narrative data, the notes that doctors write, the pathology notes, the radiology notes, contain really important information for conditions that people have. And we will be taking extracts of those, of the concepts in a privacy preserving way, like everything we do, and make that available to researchers as well. These are coming and made available through our Researcher Workbench, which we're in the process of updating. We have the beta launch of Researcher Workbench 2.0 that came out in January with our existing resource. The next data set will only come out on the new Workbench. And this new Workbench has a number of new tools. It will help investigators apply latest technologies and GPUs and CPUs. It will allow greater accessibility of running workflow technologies to speed up research, and some enhanced sharing techniques, and an even better data explorer to build those cohorts without having deep programming knowledge. We have a number of tools that we do massive compute on and pre-calculate for researchers to aid their work, and even to clinicians to help understand the patient in front of them as they find perhaps a genetic variant. This all-by-all browser is a computation of more than 1.3 trillion different pairwise analyses of looking at genetic variants and individual conditions and diseases. These data are explorable through the web, and we've done that compute for you. About 700,000 hours of compute is available, and it's available for researchers to explore and hypothesis generate. On the right, you can see a variant browser mentioned, and this is something a clinician, if they have a patient in front of them, they can look up a genetic variant and see, how common it is, how common it is in different ancestries and things like that. And then we've taken the data across all of us and another resource developed by the National Human Genome Research Institute called ANVIL, and created an imputation service. So if you have array data and you want to impute across what is the most genetically diverse population in the world, you can use that. And we show that the imputation quality using this new imputation server is better than what's available in other resources. Now, participants have generated lots of biosamples, and we take that biosample data and we want to turn it into value. I've talked about genetics. I've talked a little bit about proteomics coming in our next data release. We have a number of other resources that are doing other kinds of things. Nutrition for Precision Health. Many of your participants may be involved in some of these. For instance, that one is looking at different diets. And if you're close to one of the enrollment centers, some people are actually had randomization to diets and will actually be in a facility where they will be studied metabolomically for different diets and their effects in a controlled environment. We have generated RNA-seq data and transcription data. We'll have the long read data with methylation data coming out. And we are looking at all sorts of things. One of the ones I'm excited about in the top right there is looking at the risk of environmental exposures with type 2 diabetes. We know well the genetics around type 2 diabetes, over 600 loci that are associated. In this study, we're looking at the exposome and can integrate with people, of course, that have sequencing data as well. So we can look across genomics, environments, and risk of people who develop diabetes since they enrolled in the program. And of course, the numbers are huge with 14 million aliquots across 4 million biosamples types that people have contributed, mostly blood and also urine and saliva. But we also want to make those biosamples available to the great ideas that the research community has. And so almost any day now, we're going to release a opportunity for outside researchers to propose good ideas and use these biosamples to do new research. And so I encourage you to follow this QR code, read about this, and apply with your great ideas and how we can use our biosamples to generate new data through the appropriate approval processes, review processes, high quality data that we can generate and make available to everyone to accelerate research. Now, I want to come back to where we started and highlight a couple studies that relate from my own research lab on cystic fibrosis. First, a researcher in my lab, a staff scientist, created a population of individuals who have cystic fibrosis in all of us. Now, it's a rare condition, but still, she found 151 individuals that had cystic fibrosis, and you can see these conditions here mimic the things we expect to see. Bronchiectasis, which is a pulmonary condition, lung infections, specific infections like aspergillosis and Pseudomonas, and pancreatic. Many other conditions show up, as well as some potentially, known but maybe not talked about findings in there, all at extremes clinical significance. However, like my father, my uncle, myself, we are increased risk for having one copy, and we don't know the risk of having one copy. And there are 10 million individuals in the United States that have one dysfunctional copy of CFTR. And so we asked the question, are people who have one copy of this gene at increased risk? And there have been studies that have shown this, but typically looking at billing claim data, which is biased. People who get tested for CF have a reason for being tested, usually because they have a relative. And so what we did is we looked at people in all of us who have one copy, and in this case found about 8,000 people that have one copy of a pathogenic or likely pathogenic variant in this gene. And as you can see, it looks dramatically different. There may be some increased risk, but it's not very much compared to the general population. And so that is meaningful for thinking about precision medicine in the many, many people who carry one copy of this genetic variant. So to end, I'll just remind folks that we are finishing the day with a patient participant engagement forum. If you're a participant, please join us to connect with researchers and program leaders, ask questions, share feedback, and I will be part of that. I look forward to hearing from you and being able to answer your questions. And now I will pass it over to Dr. Naomi Allen.
So I'm Professor Naomi Allen from UK Biobank, and I'd just like to talk to you briefly around why studies like UK Biobank and all of us are so important. UK Biobank's been going on for some years before all of us, and it's really been the blueprint for how studies like all of us will also help to change health-related research, and I can guarantee will really help us to think about how we can better diagnose, treat, and ultimately prevent disease. So for those of you who are not so familiar with studies in the UK Biobank is essentially a very large-scale study, like all of us, in the UK, where we recruited 500,000 people about 20 odd years ago now, aged between 40 and 69, who lived across the United Kingdom, across different assessment centres, as you can see on the map there. We collected lots of information about them, and they gave us consent when they joined the study for us to follow their health over time, to link to medical and health related records, and to recontact them for further assessments, just like all of us. So in UK Biobank, we have a vast amount of data on all of our participants, including genetic data on everyone, including whole genome sequencing, physical measures such as blood pressure, lung function, bone mineral density, anthropometry on all 500,000. As I've said, follow-up of their health through health linkage, medical records. We have imaging data on 100,000 participants, a range of biomarkers ranging from cholesterol, sex hormones, proteomics, metabolomics, and so on the cohort. And a vast amount of self-reported data on lifestyle and other types of questionnaires. So all in all, about 40 petabytes of data that we make available to health researchers from all over the world to study the determinants of disease. So in 2017, we released genetic data for genotyping data for all 500,000 participants. And I think it's fair to say that that led to a massive increase in the use of the data in terms of how genetics can explain subsequent disease risk. Up until then, if you wanted to know about how a genetic variant influenced disease, you would have to cobble together genetic data from multiple studies to get a large enough sample size to really answer the question. With the release of this data on 500,000 participants, it overnight changed the game so that it enabled you to do really robust science to understand how genetic variability influences disease risk. And that led to the large-scale development of polygenic risk scores, which you can see on the picture on the right. When you take all of your genetic variation across your whole genome, what you'll find is that there are some individuals who are in the top, say 3% risk of developing, in this case, breast cancer across their whole genome, who have about a 25% lifetime risk of the disease. And that's equivalent to very similar monogenic disorders. And so these types of research has led to the question, well, can we use genetic data to help predict, alongside clinical data, who is at high risk of developing breast cancer or other chronic diseases over their lifetime? And now polygenic risk scores are being implemented in the UK and in the US to do exactly that, to help predict disease in the future so that patients can take preventative or early treatment. And clinical trials are also underway in anger, at least in this country, to using these genetic data to try to identify subgroups of the population who are at high risk of developing certain subtypes of disease, for example, aggressive prostate cancer. UKIP Biobank has also released whole genome sequencing data, and as Josh has just said, they're also really on the way to having whole genome sequencing data on all of their participants as well. And the value of these data is that it will help researchers to identify the biological underpinnings of how diseases develop. So this picture here on the right, the take-home message here is you can see that gene that's highlighted in the red circle there. What that means is there's a rare variant in that gene that is associated with a very low body weight. And so what research groups can do is to try to identify drug targets that mimic the effect of this genetic variant and thereby could be used as an anti-obesity drug. So you can use this targeted sequencing data to identify drug targets that will really help accelerate how new targets and drugs are developed. It can also help with what we call risk stratification, which essentially is just, can we use genetic data to identify subgroups of the population who are at high risk? For example, you could have targeted screening. Can we use these data to identify people who are more or less likely to respond to certain treatments based on their side effects profile? It could be used in all sorts of interesting ways in the future. And resources like UKIP Biobank and all of us are unique. That the amount of data that we have on all of our participants mean that this type of research is now absolutely possible. Now, one thing that's really important is there's no good finding this association, for example, between this particular rare variant associated with a lower body weight in UK Biobank if you can't then replicate it in other populations, because it might have been a fluke. So what this research group have shown here is that the association between this variant and body weight, you see it exactly in UK Biobank as you do in Mexico City studies, as you do in a US population here in the Geisinger health program. And that's really important because it's only once you have consistent associations between risk factors for disease seen in different subpopulations around the world that it can really be generalizable to all populations. And this is where it's really, I can't overstate the importance of having diverse populations, which is exactly what all of us have really tried to do from the very start of this program, and I absolutely applaud them for doing so. In the UK, we are not very ethnically diverse, and when we started the study 20 odd years ago, about 5% of the population were from non-white ethnicities, whereas all of us, just under half of the cohort, are from non-white ethnicities. And that's really important because the predictive ability of genetic risk scores may differ by ethnicity because of the differences in the distributions of genetic variants. And this is an example of just a paper that was published quite recently using the two different cohorts, basically demonstrating that performance of genetic risk scores on disease risk is really dominated by having an appropriate sample size. So we really need to have much more genetic data from ethnic minorities, and all of us will absolutely be crucial in helping us to really understand genetic differences across ethnicities. But it's not just about genetics, of course. Different subgroup of populations vary in all sorts of ways, socially, economically, culturally, diet, lifestyle, and by capturing that information as well also helps us to understand how different characteristics of our life's choices influence disease risk across the whole range of different ethnic, racial, socioeconomic population groups. So I want to talk a little bit about the imaging data in UK Biobank. All of us may well be considering doing imaging in the future, and I would strongly recommend they think about trying to do this, because having imaging data, in our case, in 100,000 participants, has been a game changer in terms of how we can predict disease early and how we can understand how diseases develop, because it literally makes the invisible visible. Here is an example of showing the amount of fat around the heart, and unique research demonstrating beyond any doubt that cardiac fat is a really good digital biomarker for heart failure. And if you can diagnose it earlier using cardiac fat, then you can treat it earlier. And it also can be used to predict who is going to develop heart failure in the next 5 years. Thereby, you can give early treatment and save people's lives. Here is another example showing from abdominal scans that you can use AI algorithms that combine these scan images that take a look at the composition of the structure and function of organs together with clinical data that can predict the risk of a whole range of different cardiovascular, kidney disease and so on up to 3 years before clinical diagnosis. In addition, the brain MRI protocol that we've developed for UK Biobank is so state-of-the-art. It can measure the structure and function of the brain, very granular metrics on brain atrophy and vascular pathology, that it's now being implemented in memory clinics up and down the country, because these automated pipelines can identify dementia subtypes much more accurately and much quicker than a consultant or a clinician, leading to increased time that the doctor can spend with a patient and increased confidence in diagnosis. So a real application of how research can actually change clinical practice very quickly. proteomics is something that UK Biobank has really been facilitating research, and all of us, I mean, Josh talked about that researchers will be able to get their hands on the biological samples, which means that you'll be able to measure things like circulating proteins in the blood. Here is an example showing that a handful of proteins is strongly predictive of different types of dementias 10 years before a clinical diagnosis, which is frankly astonishing. I think nobody thought that that would be possible. So you can start to ask questions like, well, could we use this as a potential screening tool to give those patients preventative strategies or early treatment for these types of neurodegenerative conditions? And now those of us who were really excited by genetics 10 years ago, so companies like Pfizer are now starting to say, well, proteins is actually much, much more informative than genetics in terms of identifying early disease pathology. And so this is a really exciting change for us, and it's only become about because of increase in technology and increase in the types of analytical tools that we can use, including AI, to really find how patterns of changes in proteins in our blood can identify disease early. So it's super exciting. Here's another example, and it really is just to demonstrate how you can use circulating proteins to understand how diseases develop and how best to treat disease. So this is an example. All of those proteins, which are identified by letters that you can see in red, are all the 20-odd proteins that are strongly associated with the future risk of depression. And the box at the very top is a protein that we know is involved in immune and inflammatory pathways. So that gives us a really strong indication of that is these types of pathways that are important in the development of certain types of depression. Other proteins, like the one that I've highlighted in the box below, which starts with TNF, is colloquially known as death receptor 5, which I think is fantastic. This is a therapeutic target in clinical trials for lung cancer. So it then begs the question, well, could we use that drug that's actually for a completely different condition and repurpose it for depression? Proteomics is really starting to change the game in terms of understanding disease, and can we identify drug targets that are already out there and repurpose them for different diseases. And finally, I want to end on another example of how researchers are using medical records in UK Biobank together with clinical data, but no imaging, no genetics, no proteomics, nothing fancy like that. But you can use your past medical record using AI to predict your risk of a whole range of different diseases pretty accurately. So what you can see here is the average risk for breast cancer and Alzheimer's disease on those two graphs. And you can see some people have a higher risk, some people have a lower risk. And this model can predict your individual risk of a whole range of different diseases with just as much accuracy as a single disease model. So you can see how the use of AI is really starting to change the game in enabling us to identify disease risk earlier in individuals, which could really start to transform healthcare in terms of prevention and early treatment. So finally, I just want to kind of bring it back to basics in terms of why this is so crucial to have studies like UK Biobank and all of us in different populations. You can use one cohort to make the scientific discovery, but you absolutely need the other cohort to validate and replicate that finding, especially in different diverse populations, to make sure it's generalizable to everybody. And only then can you then start to translate it into clinical practice. So my final slide is really just to summarize how cohorts like UK Biobank and all of us can advance science. And it's not just about us trying to understand the causes of disease, the genetic and lifestyle determinants of disease. It can be used to really understand how diseases develop, the mechanisms of disease, how we can predict disease early using biomarkers, digital and imaging, how we can develop new ways to prevent disease and to develop new treatments, and how to personalize medicine. There's a lot of talk about personalized medicine, but it's through studies like these that you can really do research on really how to target the right drug to the right patient and the right time. And that's exactly what we need to be aiming for. And I guarantee in the next 5 to 10 years, that will absolutely be occurring in the healthcare system, at least across the Western world. And we really need to try to build out and coordinate with other studies around the whole of the world to make sure that this type of research can happen in other populations as well. So thank you very much.
Hey, thank you, Nai. That was wonderful, and I share your optimism about the next 5 to 10 years, and congratulations on all the wonderful work you've done to bring the UK Biobank to where it is today. And hi, everybody, I'm Jeff Ginsberg, and it's my pleasure today to talk to you about the state of the science for the All of Us research program. And I'd like to frame the state of the science through the lens of our mission, how we are accelerating health research, medical breakthroughs, and how we're enabling individualized prevention and treatment and care for all of us. So the question really is how is the current state of the science advancing the All of Us mission? So let's start with accelerating health research. I think one of the key and early innovations for the program was to deliver a researcher workbench, and that was in 2020. As many of you know, the Researcher Workbench provides our data through both the public tier, which gives data snapshots and summative information, and there's access to more granular data through the registered tier and the controlled tier. And this platform is now being used, as you can see on the right, by over 22,000 researchers across the globe and over 1,300 organizations and nearly every NIH institute and center. And as you can see at the bottom right, the median time to access our data is less than 30 hours, and using the all-by-all browser that Josh mentioned earlier, a genome-wide association study that 5 years ago might have taken months or years to complete now can be done in minutes at significantly reduced cost. And we estimate that our centralized cloud data storage has saved nearly $4 billion compared to if individual institutions had to host data locally for their researchers. Now, the engine of our science is our multimodal data and scale. And you saw this slide earlier, so I won't go into detail, but I want you to recognize that not only do the counts in our data continue to grow with each release, the numbers of data types are also growing, as you can see on the right, with cognitive data, wearable data, long-read sequences, and multiomics, as well as clinical notes. So let's talk about accelerating medical breakthroughs. One of the hallmarks of breakthroughs are the publications using the All of Us data, and we're seeing about 5 to 10 papers being published a day, many in high-profile journals on topics that impact the health of Americans. And our website has a publications directory that you can see on the right that catalogs these, and we now have over 1,300 publications in the catalog. I also want to point out that we are a major contributor to ClinVar. It's a genomics variant database that is used daily by medical geneticists around the world to better diagnose rare diseases, and that has undoubtedly impacted patients' lives. So here I want to highlight a few areas in the past year that All of Us discoveries that are really poised for clinical impact. The upper left, you see a number of studies using polygenic risk scores that can be applied across multiple genetic ancestries and be used for prediction, prevention in diseases like diabetes, coronary heart disease, peripheral arterial disease. There's also wearable data that is becoming a very important marker for disease risk and prevention. And at the top right, there are a number of studies that explore genetics of exposures both to drugs and to alcohol, and that have shown that these lead to various health conditions over time. And then, as Nai just pointed out, the electronic health record alone is being increasingly used to better identify and diagnose diseases such as type 2 diabetes and the like. So let me drill down a little bit on one of these studies. This is an example of a study that showed how genetic variation in a gene called CYP2D6, which is responsible for the metabolism of commonly used pain medications, impacts pain management. Investigators showed that the genetic variants that reduced the activity of CYP2D6 resulted in increased visits to the emergency room for pain. And some pain drugs do not work well when patients are taking other drugs that also use CYP2D6 for their metabolism. And hydrocodone and tramadol were the most strongly associated and impacted by CYP2D6 variants. So the point is that the genotype really helps predict opioid response. Co-medications can create similar risk for pain, and prescribing decisions for that is, which opioid should be used to improve pain and to control pain can be improved today. So the bottom line is that both genes and drug interactions shape the opioid response, and both are actionable. So clinicians can actually use this information to better optimize pain control for patients that need it. Let's talk about enabling individualized prevention, treatment, and care. First and foremost, the All of Us data platform is the nation's precision medicine resource. We now represent not only the genomics, but the environmental complexity of the nation. We have integrated genomics, lifestyle survey, clinical and environmental, as well as wearable data at scale. There are now, as you can see in the middle panel, 8 polygenic risk score clinical tests that are now available to patients that are focused on cardiometabolic disease. And as you heard earlier, we have returned results to returned 733,000 DNA results to individuals. Nearly 550 participants have actionable hereditary disease risk, and more than 145,000 have pharmacogenetic variants that could impact medications that they're currently taking or that they may use in the future. This is a little bit more detail on what I just told you. In the left-hand panel, 3% of our participants have genetic variants that could result in malignant hyperthermia, familial hypercholesterolemia, cardiomyopathies, or cancer risk for breast, ovarian, and colon cancers. And below that, in the lower left, you can see that pharmacogenetic variants that impact the metabolism of many drugs commonly used in this country are present in more than 85% of our participants. In the middle panel shows a report from the polygenic risk score test that I just mentioned a few moments ago for scores related to atrial fibrillation, coronary artery disease, type 2 diabetes mellitus, lipoprotein A, hypercholesterolemia, hypertension, thoracic aortic aneurysm, and venous thromboembolism. And on the right-hand panel, you can see where All of Us data is beginning to impact clinical practice guidelines in obesity, diabetes, cardiovascular disease, lung disease, as well as pharmacogenetic-based drug prescribing. And I feel like this is just the beginning. So let's finally turn to For All of Us in our mission statement. And as you saw already, our program covers more than 98% of the United States, indicative of our reach to represent the United States population. And on the right-hand side, you can see the counts for major conditions affecting people in the United States. And this is extraordinary. Tens, if not hundreds of thousands of individuals with these conditions are represented in our data, which is an enormous opportunity for discovery science and translation. Now, what about the future of All of Us science? Partnered research studies, a term that we use for studies that will add new data to our core data set, are now escalating as we speak. And AI methods will be used to allow us to assemble the data that you can see in the middle panel, which, by the way, includes imaging data that we will begin to acquire later this year, and multi-omic data in metabolomics and proteomics that we will also scale up hopefully later this year or early next year. But the assembly of these different data types really is what's going to help us advance precision medicine to build better models for risk prediction, better models for diagnosis, predictive modeling for treatment response, learning about disease trajectories, health monitoring, and also the ability to discover new drug targets to lead to more impactful and innovative medications. So let me just leave you with this thought. All of us is not simply a growing data set, but we are increasingly delivering on our mission to accelerate discovery and build the evidence base for more individualized and optimal health care for the United States. And that's really thanks to all of you, and I thank you for being here today.
Thank you to our speakers for sharing your insight, and thank you all for joining today's session. I hope you can stay. We have our concurrent scientific sessions happening. You can navigate to these sessions through the agenda page, which you can find by clicking the link if you look at the top of your screen. Thank you, and I hope you enjoy today's sessions.